Hello, welcome to Medicine 2020. So, here today let us discuss a little about the parathyroid hormone. And coming to the facts of parathyroid hormone in the gland regarding their evaluation, here's a funny fact and a serious fact. The interesting fact is it has 84 amino acids. Okay, it got reduced from 115 to 84. We'll discuss it how. And the fun fact is, the first time the parathyroids appeared in course of evolution was when amphibians left the water. So they don't want to live in the water, from which they used to derive the calcium through their gills. And after they came out of the water, gills were of no use, and there comes the necessity of parathyroid glands to regulate the calcium. So this is how the parathyroids have evolved. So coming here, the sequence of synthesis of and cleavage of parathyroid hormone is from the nucleus. Okay, the mRNA comes out, synthesizes the pre-pro parathyroid hormone. It goes into the endoplasmic reticulum through carrier protein. Okay, and there it is cleaved into pro PTH, and this is transported to Golgi apparatus. Okay, and here in Golgi apparatus it is cleaved into parathyroid hormone, that is the original PTH with final 84 amino acids. Okay, with the NH2 and COO terminal. Okay, good. So this is concentrated in the secretory vesicles and granules. So this is present in the secretory vesicles and granules. So there are other similar granules which are morphologically similar, but they have here the parathormone plus proteases like cathepsin B and H. Okay, these degrade the parathyroid hormone. We will know its importance in due course of time. So this proteases, because of these proteases, parathyroid hormone secreted from gland won't have NH terminal, okay, but have COO terminal. Remember this, it is useful actually. Okay, because these are proteases and amino is very important group. This will have NH, this won't have NH terminal but will have COO terminal. Okay, and this those cleaved parts with COO terminal are called CPTH. Okay, we'll learn about this. The parathyroid gland, this responds to the levels of hypocalcium. This is very important actually. We think it's hypercalcium. It responds to the hypocalcemia more than hypercalcemia. Okay, so the absolute ionic calcium and also the rate of fall of calcium will stimulate the secretion of parathyroid hormone. Okay, this is a protective mechanism from the sudden hypocalcemia, because it will lead to tetany, right? It's not tetanus, it's tetany. Okay, so why does this actually tetany occur? So when calcium is low, it will make the RMP close to threshold. Quick firing leads to quick firing. Okay, good. So this receptor on parathyroid gland is GPCR. This is a G protein coupled receptor. It's a serpentine receptor because it makes 7 turns. Okay, fine. This is there is a calcium sensing receptor. This is a calcium sensing receptor. That means it senses the levels of ionic calcium in the blood. If it is located in parathyroid gland, renal tubules and parafollicular cells. Why? To balance with calcitonin. Okay, but remember here that Calcitonin is mostly of no use in the calcium metabolism in humans. It is in the lower mammals where the calcitonin plays a very good role. Okay, so here in humans it is mostly the parathyroid hormone. Now if there is a defect in this calcium sensing receptor, okay, if there is a defect in this calcium sensing receptor, then the calcium increase in calcium in blood, because it won't be able to sense the increased calcium, and decrease calcium in urine. Okay, this increased calcium will stop the PTH secretion, whereas decreased calcium will stimulate the PTH secretion. Okay, now if there is an activation of this receptor, this leads to the familial hypoparathyroidism with hypercalciuria. Okay, this is called FHH. This is FHH. And the vitamin D regarding the calcium metabolism has no direct action on PTH secretion, but this vitamin D, if increased, suppresses the PTH gene transcription. Okay, so it indirectly suppresses the mRNA which we discussed. Okay, and coming to this phosphate has a direct action on increasing PTH secretion. Okay, it is independent of calcium and vitamin D. Okay, it is independent no matter how the levels is. If the phosphate is increased, there is increase in PTH secretion. So that is why, as a norm of balance, okay, as a norm of balance, this increased PTH will inhibit phosphate reabsorption from kidney. Okay, and from the kidney tubules. And here, see, here I have kept this as a very important thing, alert, because in chronic kidney disease, who's a patient who's on dialysis will have decreased serum calcium and calcium calcium sensing receptor stimulation. Okay, this leads to hyperparathyroidism, but here it leads to increased phosphate but not calcium, because this PTH will act through tubules, vitamin D synthesis, and then absorption of calcium. But here there is kidney disease and vitamin D synthesis is impaired, so there is increase in phosphate. Okay, because of the bone resorption. And cinacalcet is a drug which stimulates the calcium sensing receptor, which decreases the phosphate and inhibits the direct and indirect effects of phosphate. Okay, inorganic phosphate. Here there is this is a little fact about what regulates the parathyroid cell proliferation. Here the stimulated calcium sensing receptor and increased serum calcium and vitamin D, these all three inhibit the parathyroid cell proliferation. Okay, and this is just for the namesake, if you have to know what inhibits the parathyroid cell proliferation. What is the effect of inorganic phosphate? If there is an increase in the increase of inorganic phosphate in the blood, either be it due to increased intake or decreased excretion, like in chronic renal failure, this binds the ionic calcium, okay, which is responsible to stimulate the CSR, and also it directly stimulates the parathyroid hormone. Okay, this increases the levels of PTH and decrease the calcium excretion. Okay, this is one of the very good mechanisms. And added to that, how this calcium is absorbed in the kidney is very important for understanding the homeostasis. Okay, when there is calcium that is filtered from the glomerulus in the PCT, in the late PCT, it is absorbed in a paracellular pathway along with sodium. Okay, and this creates a gradient where when it reaches the thick ascending limb, here also it is transported in the same paracellular pathway along with the along with the sodium. And here in this same thick ascending limb there is calcium sensing receptor. Okay, see this magic. The same calcium sensing receptor is present in thick ascending limb, regulating the absorption and excretion of calcium. But here in DCT what happens is, usually sodium and calcium absorption are independent, but when thiazides act, this increases calcium absorption and promotes the sodium excretion. So that is why it is used in stones, renal stones with high calcium content. Okay, and this is how it is done. Actually here the around 60% is absorbed, around here is 40%. Okay, it is absorbed around 40%, here 1% and 1% in the collecting tubule and all that. Okay, but here look at the action of parathyroid on these tubules. Parathyroid, what it does is, when it is high, that means the serum calcium, the parathyroid when high will inhibit the absorption of sodium and calcium in the PCT. Okay, see here, sir, it should increase the absorption, but how come it is decreasing? Okay, but what it does in the next part, see what it does in the next part is, there is less stimulation of CASR, right? And so this PTH, increased PTH, will stimulate the absorption of calcium from the tubule, the late thick ascending limb, and also the distal convoluted tubule. So out of this, if we look at here, one CA is inhibited but two are absorbed. That means net is increase in calcium reabsorption. Okay, added to that, it also inhibits the reabsorption of phosphate. Okay, which is very important. That is why in PTH increase there is more ionic calcium and low phosphate. Okay, and what is the effect of body pH on the calcium? See, in acidosis at DCT, the calcium absorption is inhibited. Okay, inhibit the membrane apical channel. Acidosis will inhibit, the pH will inhibit the membrane apical channel, whereas alkalosis will stimulate the same channel and promote the absorption of calcium at distal convoluted tubule. Okay, but here look at this. This is very, very important. Okay, because the protein, the calcium that is present in the body is in many forms. Okay, it is in the bone, it is bound to protein, it is in the ionic form, and others bound to the phosphate, HCO3 minus, and etc. Okay, but here when there is acidosis, there is more of H plus, right? So this H plus will occupy the albumin, leaving behind the calcium. So there is increase in free calcium, whereas in alkalosis H plus is less, so calcium will bind to the albumin and there is decrease in free calcium. Okay, but here there is a small point you can notice. See, if there is magnesemia, hypomagnesemia, if there is magnesium, hypomagnesemia will act like hypocalcemia. Okay, so thereby increasing the PTH secretion. Okay, this is a small fact to remember for you. And followed by that, now coming to the action of PTH on the bone. So we have seen how it acts on different levels, how it is secreted at different places and all that, right? So when there is an increase in parathyroid hormone, it stimulates the osteoblast to produce monocyte macrophage colony stimulating factor. Okay, and also a rank ligand. Okay, what is it of help? So what is the need of this? This MCSF will bind to the receptor on the lineage precursors and make them differentiate into pro-osteoclasts. Okay, this rank ligand will bind to this rank receptor and promote the, it is not just a single cell, right? Osteoclasts, many pre-osteoclasts will be many. Okay, this will promote their fusion, leading to giant multinucleated osteoclast. And there is OPG. Okay, it's a it is also a protease like thing which inhibits the binding of rank ligand to the pre-osteoclast and thereby stopping the differentiation. Okay, these osteoclasts will bind to the bone and create an airtight space. Okay, they bind very tightly and create the space and release the hydrochloric acid, HCl, and other proteolytic enzymes, because the bone is composed of organic and inorganic materials, right? So it needs both these hydrolytic enzymes and everything, and leads to bone resorption and thereby increasing the inorganic phosphate and calcium in the blood. So this is how it happens. It is a fact to remember that PTH acts on osteoblasts, not osteoclasts. Okay, increases the rank ligand, increases MCSF, and thereby increasing the osteoclastic activity. Okay, at lower levels there is we'll discuss it. We'll discuss the PTH. The concept of measuring the intact pH. See, when there is hypercalcemia, there is increasing calcium. You are suspecting increasing PTH. Okay, so what do you want? You'll simply order the measure of PTH to some lab XYZ. They will give you a value of 1000 and you panicked, do all the scans, find out nothing, right? So why this happens? See, that is how there is an evolution of measuring the PTH. Okay, that is the intact PTH now. See, when there is decrease in the blood calcium, it promotes the PTH release from gland. Okay, how? This decrease in blood calcium level will inhibit the intracellular degradation of this PTH hormone, and thereby it releases the intact PTH outside, that is with 84 amino acids. Okay, with NH2 and COO terminal. Okay, and whereas there is increase in calcium, it promotes the degradation of PTH in the gland and making it into small fragments. These will not have the NH2 group and terminal will not be there because they are proteolytic enzymes. Only COO will be intact. Okay, and this COO has many other biological actions which are not necessary right now. This they are called CPTH, as I told earlier, right? They have very individual biological actions. Okay, previously it was thought that CPTH has no action, but it has action in various disease pathogenesis and everything. That's not so important for us right now. Whereas here in PTH metabolism, if for suppose 100% PTH is released from the parathyroid gland, 70% is metabolized by the liver. Okay, 70% is metabolized by the liver. For suppose this is the gallbladder, just think of it. Okay, and 20% is by the kidney. Okay, and 10% is intact. So now our word came intact. So what are these? Are they not intact? No. These those metabolized by the liver, okay, and the kidney, they are called fragmented PTH. Okay, and they are excreted in kidney usually. Here there is a small difference between those metabolized by the liver and kidney. Those metabolized by the liver will not have an NH group, NH2 terminal. Okay, whereas those by kidney will have NH. And coming to that, this is not a significant difference, but just to know it, because ammonia urea cycle is produced in the liver, so it will not allow NH2 to escape. Okay, and this intact PTH, finally it produces some biological action. Okay, so fine, now you are suspecting a PTH increase. And what is intact PTH measurement? It is now the gold standard, or currently the trending measurement of the PTH. So old PTH analysis had to detect an epitope which is non-specific. Okay, that means it is present in both intact and fragmented PTH. It was not so developed, all these immunochemistries, all that. So in the new intact PTH analysis, this detects the PTH exclusively which has both COO and NH2 ends. Okay, and here now what you will know is the perfect complete PTH, okay, which exerts biological action. Okay, that is the PTH you want to know the level for. And coming to the final fact, and at low levels PTH exhibit osteoblastic activity. okay, at very low levels. This has been used in the treatments for various diseases like osteomalacia. So low levels of PTH and PTH analogs, low levels. Okay, so this is how usually the PTH is regulated. Okay, so main hormone for calcium regulation is parathyroid hormone, but not calcitonin. Okay, for that phosphate regulation and all that. So now you know what PTH is for and how it acts. So it is very simple. It looks like a bizarre thing, but what we went through all this time is usually informative because it is highly segregated. PTH has multiple options. I mean, you know, multiple actions at different sites. So it seems a little dry, but it's very conceptual if you look at it properly. So that is all for this class. Thank you.